Welcome to our spring term semester. Great to have everybody here. If you haven't got any focaccia, please eat some. And I'm sorry if I didn't make enough. No, we're good. Okay. Welcome to Lee and Shuwen that we have online from the Land Institute. They're going to be speaking today with our cereals breeders, Hannah and Jim. Let's see, I have to remember what I say at the beginning of these meetings. Welcome, everybody. I want to start by acknowledging Forever Green's funders, the Environment and Natural Resources Trust Fund, the Minnesota Department of Agriculture, Clean Water Land and Legacy Amendment, Builders Vision Initiative, and the McKnight Foundation. Thank you to our funders that make this and much of our research possible. Reminding everyone that these videos are recorded and it'll be uploaded to our YouTube channel. If anyone wants their faces to be blurred or anything, let us know. And if you're online and don't want your face to pop up, just turn off your video. Okay, Mitch, any announcements?
I do. So first of all, welcome back. New semester. The world's in rough shape, we can all acknowledge, but it's good to be focused on work that is hopefully leading to real meaningful change in the world. It's also a little surreal to be working on stuff that, is sometimes on a decadal time scale before we're going to see that change. Well, there's a lot of really intense stuff happening in the immediate, but I think we can hold that hope together that we're doing something that will make the world a better place in the future. So I'm glad we're all here together to do that together. Other announcements, the legislative session or process is kind of starting to ramp up. We've had a couple of meetings, Commissioner of Agriculture this week, Senate Ag Committee Chair this morning, all really good. And there's not a ton at stake in this session because it's not a budget year. So it's a little bit more of keeping our friends in the legislature informed of all the great stuff that we're doing and making sure that everybody's kind of up to speed and ready for next time when it will be a budget session. And there'll be some hearings. They won't be kind of the big blockbuster hearings where we have like half a dozen testifiers and that sort of thing. So TBD if there will be testifying opportunities, but I'll definitely share the details as far as if people want to come listen and be part of that. And, if any of your representatives or senators have to be on the committees, say hi to them. It's always a good thing. So I'll keep you posted on that. We are hiring a winter oilseeds specialist. So this is a position that we have wanted for a long time because there's so much work happening kind of in the interconnecting spaces between the research and the market and policy. And at times Colin Curitan has led that work. At times I have led that work, or we have tag teamed it. But we just don't have the capacity to do all the follow-up and run down all the leads and create all the opportunities or, actuate all the opportunities that are out there. So this role will be very multifaceted, somebody who needs to be able to understand the practicalities of production and a bit about the research and also be able to speak policy language and talk to market partners. So as usual for many of our roles, we're looking for that unicorn person who can do all those things at the same time. But I'm optimistic that we'll find somebody good. And so if you know anybody who maybe they've got a good ag policy background, maybe they have worked in ag industry and they're also really excited about protecting the environment, or they did a master's and now they've gone to industry, so they have those two perspectives under their belt. Those are the sorts of people who might be great fits. I want to let everybody know that a state-run program that supports our market partners, it's called the Developing Markets for Continuous Living Cover Crops, just recently announced the fourth round of recipients. So we've now, through this program, put $2 million of state money out to partners who are building businesses to advance our green crops. And that's really exciting. And the last thing, this is like the most announcements I've ever given, but I actually thought about it ahead of time this time. So in the most recent executive committee meeting, there was a call to be more transparent and let the community know what is discussed in our executive committee meetings. So first, many of you may not know we have an executive committee. It is primarily focused on our R&D program, and it is made up of Nick, myself, Colin, and Anne, the directors, quote unquote, as well as 5 to 6 elected faculty members. So Jess and Josh and Jake are all in the executive committee, as are Georgia Noor and Kevin Smith, and we used to have Praveen before he left. And so we meet every month to discuss matters related to the R&D committee, or to the R&D program, including the grant program. So one of the things we discussed in our last meeting, which was last week, I think, was just reflecting on how the last grant program process went. We've done that a couple of times, and now we're starting to shift to thinking about how can we improve for the next time around. Very important. And then we're also developing a plan for turnover among the members of that committee, because the idea is not that these are seats for life. Originally, there were 3-year seats, and that 3-year mark is coming up. And so now we're trying to figure out a system for kind of a phased turnover, so we don't lose everybody at once and have to restart. But figuring out exactly how to do that, how do we have people gracefully exit and then new people be part of the next election? Those are the things that we're working through. Okay, I'm done. Anyone else want to share updates?
I could mention the Perennial cereals breeding position closed January 5th. The applications closed, and we had 62 applicants, and we're in the process of reviewing those applications now.
This was kind of alluded to in one of the recent weekly newsletters. We have a weekly newsletter now. Yay! But we also had our co-director nomination process and application process not closed on January 19th. And we had 5 applicants. So I feel really good about that. And it was an internal candidate pool of existing University of Minnesota faculty members. So a pretty small pool, so that we have the fact that we had 5 people who are interested in upending their lives and devoting a huge amount of time to us is, I think, a good validation. So now I think I'm really done.
Amazing. I think it's fine to have extra announcements after such a big break. We have a lot going on. Okay, I'm going to stop sharing this. Hannah, you can go ahead.
Hi everyone, my name is Hannah Stoll. I'm a postdoc here at the University of Minnesota, currently doing perennial cereals breeding and genomics. Jim Anderson is in the room as well. I'm going to start out by talking about perennial cereals breeding at the University of Minnesota. So today we're going to have me speak for 20 to 25 minutes, and then also on the call is DeHaan and Shuwen Wang at the Land Institute, and each of them is going to talk about intermediate wheatgrass, which is Lee. I think we'll have Lee go right after me, and then Shuwen Wang will talk about perennial wheat at the Land Institute. So it's going to be a relatively quick talk for each of us, with the hope that we can all talk together at the end. But I'll hopefully have at least a couple minutes after my talk for a couple questions before we move into Lee. So I'm going to start off by talking about intermediate wheatgrass breeding at the University of Minnesota. So a quick history lesson. I'll just start with the history of intermediate wheatgrass breeding at the University of Minnesota. We received our germplasm in 2011 from cycle 3 materials from the Land Institute. And since 2011, we've completed 9 breeding cycles, and we're entering breeding cycle 10 here at the University of Minnesota. And starting with a few cycles ago, we are doing 1 breeding cycle per year. Prior to that, we were doing 1 breeding cycle every 2 years. So we're increasing the speed too. Notable variety releases. The first one was UMN Clearwater. And then our most recent variety release, which I'll talk a little bit more about, is UMN Itasca. So our breeding priorities for intermediate wheatgrass or Kernza here at the University of Minnesota. There's a lot of them, but grain yield per head and 1,000 kernel weight are most notable, I would say. Free threshing seeds, so seed that comes out of the hull more easily off the combine would be ideal. Non-shattering types, and some two I guess relatively new initiatives are breeding for better seedling vigor and persistence of yield over multiple years. I'm not going to go into the details of like how we'll be doing each of those, but in the questions, please feel free to ask and we can talk more. And we also track spike traits like length and width, plant height, lodging, general disease resistance as well. So fusarium iron head blight, leaf rust, things like that. A little bit of I guess just an update on varieties that are in development. So as far as released and near released varieties, MN Clearwater released in 2020. MN Itasca was technically released in 2025 and seed will be available in 2026 and 2027. And we have another number of other varieties that are in our pipeline. So MN 19s and 22s are undergoing yield testing and some seed increases. We have some additional varieties that'll be entering yield trials beginning this year. So those will be planted this fall. And as a breeding program works, we always have additional synthetic cultivars that are being created and going into yield trials and further testing and seed increase. So it's a pipeline. There's always more coming. So a question that we've been getting a lot and I wanted to briefly talk about was this new variety, MN Itasca. What can we expect from it versus Clearwater? So this figure is going to be part of a publication. Praveen Bashkane is the lead author on this publication. This is his figure, but I'm presenting it. But this is a good visual for comparing how MN Itasca shapes up in comparison to, or shapes out in comparison to some other experimental varieties and MN Clearwater. So just for sake of a simple visual, MN Clearwater is in the light pink. MN Itasca, which is our latest variety release, is in the yellow. Across all metrics, MN Itasca has incremental improvements over MN Clearwater. That's the punchline here. These data were from 12 different environments collected between 2021 and 2024. A little bit more details about other yield trial results. This particular figure is from a yield trial that was grown at 2 locations, and I have 3 years for St. Paul and only 2 years for Lamberton. I think the gist here shows us how MN Itasca shakes out. So I should probably explain a little bit more what's going on here. The variety is on the x-axis, and I ordered it by mean yield in descending order, and then the yield, which is adjusted based on our model, is on the y-axis. So this is just a ranking based on yield. As you can see, there's not really statistical differences between each variety candidate. However, you can see the rank for what that's worth. Another important takeaway here is we had some TLI varieties grown in these Minnesota environments, and the question we've been getting from growers is, hey, can I, what are TLI varieties? Can I grow them in Minnesota? And basically what these data show is that you can grow TLI varieties in Minnesota and it most likely won't be a total disaster. And then I also have the results from the most recent yield trial. This is in yellow and red because it's only 1 year at 2 locations. And anytime I present this to growers, I want them to know, please don't just take this out and say, okay, I'm going to plant X variety because of this one figure that I saw one time. But all this to show we have additional variety yield trials that are ongoing. MN Itasca and then Clearwater in those variety trials. It'll be interesting to see how things shake out after an additional year or 2 of data. And yeah, we have some other TLI varieties of this variety trial as well. And TLI and UMN, we're already talking about collaborating for better germplasm exchange between the 2 programs and testing each other's varieties and each other's environments.
Becker's a pretty tough site for Kernza too, it looks like.
Yeah, I think part of this was planting was planted a little bit late, so that's a consideration. But yeah.
DeHaan,
Yes.
what's MN Itasca's research name? What was the number?
Oh yeah. So the question was, what was MN Itasca's research designation? It was MN 1603. I think I covered that. I just covered it up for goodness sake, but MN 1603 was its research designation. So MN Itasca came from cycle 2 of our breeding program. Yeah, and we're currently on cycle 10 for that information. Yeah, Jess.
Does Becker usually have worse yields, or do you think it was more of the planting date than the?
We don't have a whole lot of data coming out of Becker actually, because we just started planting a lot of our breeding program nursery there 2 years ago. Largely, it's a different soil type, it's a sandy soil. It's easier to manage because it's closer. Lamberton was undergoing some like staffing changes a couple years ago, and that was, I think, why Praveen moved to growing our breeding nurseries in this field trial in Becker, just out of those logistics, and then it also is just easier for us to manage more intensively because it's a day trip versus an overnight trip. Typically, they're a really long day if you go to Lamberton.
Is that variation about the mean for St. Paul normal?
Hopefully not. I don't know. Okay, I'm gonna keep going. Sorry to blaze through. The gist here, our breeding program is entering cycle 10. We're doing 1 cycle per year using genomics assisted recurrent selection. Itasca is incrementally superior to Clearwater for all the recorded traits in Minnesota environments. And future breeding work, we're going to prioritize grain yield, of course, the persistence of that grain yield, seedling vigor, and improved threshability. We have a lot of varieties in the pipeline, and several are undergoing seed increase, and additional yield trials will be established this coming fall. Okay, that was intermediate wheatgrass. It's 12:20, so we're doing okay on time. So I think we started about 10 minutes ago. Yeah. So the next group of things that I'm going to talk about are perennial cereal rye and perennial wheat breeding. Okay, so why are we interested in breeding perennial cereal rye in the first place? And it gets back to this dual use question. So used as a forage, you can see early season, rigorous early season biomass production. This is just a direct comparison of Kernza versus perennial cereal rye. I'm not necessarily suggesting perennial cereal rye as like a replacement of Kernza, but just for people who are familiar with Kernza, this is what the difference of biomass looks like. I don't need to sell the ecosystem services there. And it also has promising grain yields as well. So pictured here are annual cereal rye, PC rye, perennial cereal rye, and Kernza seeds. We've estimated around 50% of annual cereal rye yields, and we had an initial seed increase, variety trial, whatever you want to call it, on Kurt Kimber's farm, and he estimated 37 bushels per acre, which is really promising. So where does perennial cereal rye come? It's an interspecific cross between annual cereal rye and perennial wild rye. So how this works, you have your annual crossed to your perennial. They're repeatedly back crossed and selected for perenniality and other agronomic traits as you go through. This was originally developed in Germany in the 1960s. Sorry if it feels like I'm going fast. It's because I am. So the other consideration with perennial cereal rye breeding is that there are diploid and there are tetraploid populations present. So we have varying ploidy, depending on what you're talking about. It's thought to be primarily outcrossing, but we really don't know the degree of selfing either. So that's something that we're also trying to figure out this spring in our in some greenhouse experiments. Getting back to the history of perennial cereal rye breeding at the University of Minnesota. So in 2020, Praveen Bajgain, he was who started this breeding program. And I actually remember as a new PhD student at the time, him talking about how excited he was about perennial rye. Like, this could be the next big thing. So credit to Praveen if this really is the next big thing, because he was excited about it long before anybody else was. So in 2020, around 600 plants were planted in St. Paul, and this nursery comprised of some diploid, tetraploid, and strictum, which is like another wild perennial rye exceptions from USDA. The only reason my understanding of why he did not plant S. montanum was just because there wasn't any germplasm available. These were evaluated for multiple years. And the big takeaway from these evaluations is that seed shattering was an issue. And determining true perenniality was also another issue. It was hard to tell in this nursery what was from a shattered seed and what was a true perennial type. So cycle 2, we began selecting more stringently for perenniality. Not going into a whole lot of detail, but we had some basically repeated greenhouse fertilizations to simulate multiple winters. We also had a seed increase planted at Rosemount that was mowed off for multiple years. With the goal of selecting for perenniality. And cycle two plants were transplanted in 2023. And unfortunately, in 2024, we realized perenniality is still an issue in this population, despite additional selection or more stringent selection for perenniality. yeah, so flag some of these plants. But we have a plan for that. So
I'm sorry, did you flag the ones that showed perenniality or?
yes, yep, yeah. Thanks. So we have a plan for that. There's kind of two paths that we're forging going forward with the goal of we need to select stringently for perenniality, while also making additional crosses, selecting for agronomic traits, things like that. So in the field right now, we have three replications of parents that were selected from genomic selection or using genomic selection from a cycle 2 population. And that field experiment is going to be used for assessing these agronomic traits. One of those reps will also be mowed off to ensure that any plants that are in the field are actually the perennial type and not just a shattered seed that came up. And then we also have several seed increases planted. And these seed increases were derived from a population that was planted at Rosemount and mowed off for multiple generations and not allowed to, for multiple years and not allowed to go to seed. So theoretically, this should have undergone even more stringent selection for true perenniality types, and might be able to select some individuals from those populations as well over multiple years. Okay, other considerations, perenniality is the big one, ergot, seed shattering, several economic traits. And the other big question that we kind of need to answer going forward is the ploidy of our population, what we actually have. So this spring I'm planning on doing flow cytometry to basically figure out what the ploidy of the individuals in our population actually is, and probably choose one or the other and move forward with it. In summary, we also have 4 synthetic variety candidates that are currently undergoing seed increase and testing. We're on cycle 3 of the breeding population. We have multiple paths for how we're going to simultaneously select for perenniality and agronomic traits. And in 2026, I plan on initiating 4 to 6 more variety candidates. So that was perennial rye. We also just initiated a perennial wheat breeding program. So moving gears to perennial wheat. So perennial wheat is also an interspecific cross, similar to perennial rye. You have but it's intermediate wheatgrass, which is Kernza, crossed by an annual wheat, so hexaploid wheat or durum wheat. At the University of Minnesota, in 2020, we actually had some basically field trials of germplasm from the Land Institute and Washington State grown here. Those were grown for multiple years. And in 2023, Praveen selected around 24 of the best looking plots, harvested those and replanted them in 2024. And then in 2024, he selected 6 different lines that were superior for plant height. They had fertile heads, head sterility is an issue. They were earlier maturing and looked mostly disease-free. So that was the beginning of our breeding program, was these 6 genotypes that Praveen selected along with some other germplasm from the Land Institute. And just starting in December, moving into January of this year, we started crossing that germplasm in the greenhouse. We've had relatively low seed set, but not 0 seed set from what I can tell, which is the important factor. So next steps for this are selfing that F1 seed. My goal for the fall is to be able to plant some head rows. And those head rows we'll evaluate. We'll do some additional crossing depending on a number of factors. And another kind of next step with the next round of Forever Green Initiative grants was the Youngers Lab is actually going to be testing additional perennial wheat germplasm at multiple locations. And the idea with that is they'll be testing additional germplasm, and anything that it looks like it's doing well in Minnesota can then feed back into the breeding program for additional crossing. So continuous germplasm improvement, but we've already started the crossing process too.
Is self-incompatibility recessive?
There's a number of genes. I don't know if I would call it recessive. What?
Well, just if you're gonna self the F1 seed, do you think you're gonna get seed from that?
Yeah, it'll probably be variable. Yeah. So there's a number of self-incompatibility loci in intermediate wheatgrass, like several. I spoke too fast.
Simple question: as you made your perennial wheat crosses, did you just make every possible cross among those 6 lines?
So yeah, we have more than the 6. Yeah, Bren's shaking his head. Bren is the one who's been doing a lot of this. It's kind of a whole lot of whatever's flowering at the same time. We're making like every possible reciprocal cross that we can. So
Okay.
Were you able to save pollen to use later?
No, we haven't been doing that. We staggered, so we planted multiple reps and we staggered when we put them in the fertilization chamber and when we brought them out. So we've had basically almost 6, 7, we'll probably end up with about 8 weeks of just progressive flowering. So theoretically, if we wanted to make a cross between genotype A and F at any given time, we'll typically be able to make that. Things are synchronized. So
I have another question about testing for perenniality in perennial rye. I'm not nearly enough of a physiologist or developmental biologist to know the answer to this, but I'm wondering, are we sure that mowing after flowering is going to really replicate what the plant experiences when it goes all the way to grain production? Or is there a point where
no is the correct answer. No.
Okay. We're doing the best we
Does
can.
somebody have a does somebody have a so the comment was basically like, if you just mow after flowering, does the plant really think that it's, was that enough physiologically? My answer is I don't know. And I think that so we didn't, sorry, I like kind of zoomed through it, but we did 2 kind of synthetic winters, I guess, with vernalization chamber was one way of selecting for perenniality. And the short answer is I don't think that was good enough to make the plant think that it was actually going through 2 winters. But I don't know what sort of physiological thresholds are enough. But
wouldn't this be a pretty conservative method for selecting for perenniality, because we might accidentally be killing plants? So here's what I'm thinking is that if you mow it and you don't get regrowth, could that have happened because of the mowing process? Like, could that genotype have been negatively influenced by the gene? So probably.
We're selecting the hardiest,
Yes.
most definitely perennial
Right.
plants.
Theoretically.
Yeah. That makes sense.
Could use the pre-emergent.
That's what we are thinking about, and we're probably gonna test that this year.
Like a low plot and then a pre-emergent as well.
Yeah.
I suppose.
To do what?
To pre-emerge, prevent the regrowth of the seeds.
Oh, yeah, I was
I
wondering.
think
about
Brett
that too.
actually did do that in the perennial cereal rye nursery, like between years. So that would have been like 2023 to 2024. And there's still, like, even with the pre-emerge, it was still,
Yeah.
there was a lot of seed. Probably not as bad as it would have been,
It's
but
not that.
yeah.
Yeah, I was wondering that too, because it seems like whatever you're doing, you're still getting some winter kill. So yeah, it seems like some way to prevent the shattered seeds from coming back.
Well, it depends too on
Yeah.
when do you apply it and how much does it rain?
Yeah.
Yes.
Very good. It seems like a good thing, right? To select for forage, like things that can be harvested multiple times or raised multiple times.
We have a question online from Pete LaFontaine. Are perennial and annual rye roughly equivalent nutritionally?
We don't know. That's a question that the next round of evergreen funding hopefully can give us some more insights into. So Georgia Nor will be looking at perennial rye seed.
And I'll just say a little bit more about that. So kind of, I don't know for sure if this is going to be under the umbrella of that new grant or not, but the commercialization team, which none of them are here in person, so I'll speak for them, I guess. If anybody's online, feel free to chime in. Is thinking about, okay, we have a new crop that seems to have some potential agronomically. What should we be doing with this brand new crop as far as laying the groundwork for commercialization? There could be an impetus to say, hey, it yielded really well on this one farm. Let's go plant it on 10 farms and we'll get a bunch of it and then we'll try to sell it to somebody, like we're trying to not follow the frenetic path and instead be more methodical. And so from the production that occurred on Kurt Kimber's farm, the plot that Hannah mentioned, I think we got about 900 pounds of grain off of that. So we'll use a small fraction of that and partner likely with George and potentially with some outside labs as well, TBD, just to do a basic kind of compositional analysis. What are we working with here? Protein, carbohydrates, etc., oil profile. And so that will be the first step of answering your question, Pete.
Oh yeah, so Lee made this is regarding Matt's question kind of. So self-compatibility in the hybrid perennial wheat is generally dominant. He said bad seed set is mostly coming from lack of genomic stability. So chromosome loss. Not specific. Thanks, Lee.
And then we just had a question about the mowing again from Carmen. What stage of growth was the rye mowed off? Early, late green stage?
Post anthesis. Post anthesis. So after flowering, but before seed set.
Yeah, so here another whirlwind tour through some of what we've been doing at the Land Institute. I'll focus a little bit on the variety development stuff. Early breeding here at the Land Institute was done through just phenotypic selection. We grew out thousands of plants, measured traits on individual space plants, and then picked the best ones. Mostly on seed size and yield per head, and did that for about 5 generations. And as Hannah mentioned, the cycle 3 was the parent of Minnesota Clearwater, and kind of a selection out of that cycle 3. So very similar to cycle 3, but a little bit better. So about like cycle 5 in many comparisons we do. And you can see here that over time we were shrinking the timeline of each generation. So early on, the work done at USDA in New York was 5 years per generation. I went down to 3 years and then to 2 years. And at that, if you just extrapolate this out, we can figure that about 60 more years at this rate to roughly match yields of wheat in Kansas. So the idea is to try to find ways to get through more cycles faster, to reduce this time from 2 years. Initially, I went down to 1 year. And the approach is genomic selection. Hannah talked about it. We won't go through it at great length again, but the basic idea is in this green box, to have field evaluations. These can actually be in plots where we have rows or solid seeded plots or space plants. We can do them at many locations over many years and associate that with the genetics to make a model that we can predict. And then we can run this genomic selection on the right-hand side. And in just 1 year, we were doing a generation a year for several generations. And now we've gotten it down to 2 generations per year. I'll talk a little bit more about that. Now 6 months. So lots of plants. We genotype about 4,000 plants per generation, so about 8,000 per year. And then I worked out some techniques to make heads a lot faster. So I accidentally discovered that putting them under this far red light, we could get them to head without 6 to 7 weeks of cold treatment. So we cut off at least 2 months off our breeding cycle. A few other tricks I've gotten it down to. We can do a generation in 6 months or less. What does our model look like? So we've been just adding more and more data to the model over time. So we don't start over with new training every cycle. We just keep on building on all the training data we've had in generations past. So this is like kind of a summary of how we had over 7,000 genotypes, and then in a bunch of different environments as well. For every year we've been testing, and you can see here that half the variation is environment. So that what the weather is like generally is driving a whole lot of what's going on. The genotype itself is about 18% of the variation. That's the additive genetic relationship. And then we have 7 or 8% the interaction between the genotype and the environment. The bottom one's kind of a dominance measure. So, and we also use a spatial effect model. The heritability, we roughly say, is about 0.38 for this trait of yield per head. We then calculate these predicted values for each genotype. And if we just run this again and see how accurate the predictions are, they're usually over 90% accurate. Okay, how's it going over time? So this goes from cycle 6 to 14. We're currently starting 19. But This is the trait of percent naked seed. We really started to push heavily on that around generation 6 and 7. Kind of maxed it out, and so I've basically relaxed selection on this trait because we can always find plants that are 99 to 100% free threshing at this point. So it's diminishing returns now. We increased it from 50 to 80% in just 6 years. With the phenotypic selection, we're kind of going at half that rate and change. Not surprising, we're doing twice as many generations for the same time. This trait of spike yield, here it goes up to cycle 18. These earlier generations here, we don't have training data from those years included here. So I think what's happening is we're not predicting them very well because there's so many generations back. So it looks like they're staying the same. In fact, that we were making a lot of progress back then. But the model is much more accurate for the current generations because it contains the recent generations data. Current varieties, so cycle 3, as I said, Clearwater came out of that. Cycle 5 ended up passed around from people and ended up in Russia, and they asked if they could make a variety in Russia, and that was released about 10 years ago. And then we had a series of, out of cycle 7, the 701, 3, and 4, a lot of these were used in the Kerns of Cap trials. And then 801 was out of my 8th generation. This was sent to Ukraine, and they've done a lot of trials there and proved that it's far better than their other intermediate wheatgrass varieties that they have. It's native to that country, and the forage breeders, but also they're breeding for grain in Ukraine with collaborations with us. So they found excellent yields. They're typically getting yields around 1,500 kilograms per hectare in Ukraine. So it likes it where it came from a lot. Hopefully that holds up over longer periods of time. Yeah, so kind of as Hannah said, much the same traits. I put an asterisk by non-shattering because I've discovered that there's a pretty strong positive correlation between shattering and grain yield. So if you have a lot of grain, then it can shatter. If the plant is mostly non-fertile, then it doesn't shatter because there's no seed to go on the ground. So it's really important to watch that. If you start selecting for something that's negatively correlated with what you're trying to achieve, you might go backwards. And I've seen some evidence of that being an issue. So currently I'm basically allowing shattering to increase slightly over time because it's associated with yield so strongly. Here's a variety trial that is from the second year, just for simplicity to show that. You see a lot of the Minnesota lines there on the left, 19, even 22, that's from their year 22 breeding program. Minnesota Clearwater here in the middle, a lot like C5. And then if you look towards the right-hand side, you see TLI 801, which was kind of the best variety that a lot of farmers are currently using. But then, so 1102 has consistently improved, and over C5, it's almost a 50% increase in grain yield. In this trial, here's a new trial that we had first year data last year. There was even a 63% increase there. So it's looking very promising. It's pretty clearly superior to TLI 801, so it's very exciting. It's about a 20% improvement over 801, the current most popular variety farmers are growing in Kansas. I'll just call your attention to TLI 1001 here. You see, just because they're newer numbers doesn't mean they're going up. So you make these variety candidates and you hope they're better, but that's why we test them, because they aren't necessarily always. But also look at why this might be. On the next graph, we'll have the same varieties, and you see that TLI 1001, this is maturity. It's a very late maturing type, and Minnesota types, Minnesota Clearwater, very late maturing, and a lot of the difference between performance of Minnesota varieties in Kansas, I believe, is due to their late maturity. If they don't do as well, it's because they're trying to fill their grain when it's getting extremely hot here in Kansas. So this 1102 is a great yield increase and also much earlier. So I'm really pushing hard for earliness in the program now. Kansas wheat is about the earliest in the world, and the reason that we can grow wheat here is because it's so early. It just has to beat the heat. Okay. So there's more things coming down the line. As Hannah showed to you, it takes a long time from the breeding cycle. So the farmers are currently growing cycle 801 materials, and as you saw, 1102 it's currently in seed increase on a farmer's field, and then I've got varieties in the works out of cycles 12, 14, 16, and 18 as well. But before we have data, it's going to be another 5 or 10 years for those varieties to come out. Okay, quick summary. Genomic selection really helps us to go faster, but just we can't expect all these variety candidates to be better. Many varieties have great potential for higher yields, but there's lots of ways for yield to go wrong, and we try to be careful about telling farmers that the environment is more important really than the variety you plant in most cases. So you can plant a new variety, and if it's not handled properly or you get really bad weather, it's going to respond mostly to that. So looking at that 60-year timeline, if we can continue to go basically 4 times faster, the variety data also show that we're on track to hit about wheat-like yields here in Kansas in another 15 years or so, at least out of the breeding program, and then got to get to farmers.
Yeah. So if you're seeing that fast of yield increases, are you still trying to select for shorter varieties too to hold up those big seeds?
I've never found a correlation between shortness and lodging, except that the shorter plants tend to lodge more. So that's been frustrating. Basically, short plants tend to be just weaker, wimpier, poorer rooting, and they don't hold up the plant as well. So so far, we haven't done a whole lot directly on. I've tried really hard, but for the most part, the taller plants are better in almost every way. I do see some evidence now coming out that maybe we can find some reduction in height, and it's partly because the earliness tends to be associated with smaller plants. So you're earlier if your head doesn't have to sit there and elongate for 3-4 more days, and so the earliness is tending to make them a little bit shorter. So it's been a nice roundabout way, because otherwise you just measure for short plants, you just get wimpy plants. So I think maybe we got a little bit of an avenue there, but in general, lodging is not a huge problem here unless you get a tremendous thunderstorm. So it's basically not a priority trait at the moment. I saw another question here online. Is it high heat during pollination and grain fill determines environmental potential, or moisture, or both? Yes, probably both. Moisture is probably a bigger deal overall, but when it comes to targeting what we can do through breeding, it's mostly making them earlier, and that is probably not as much pollination. It's a lot more the grain fill stage, so it seems to pollinate pretty darn well. I think it's more the heat stress that's happening during grain fill, making them run out of moisture, so burning through the moisture. So it might be an interaction there between, it gets hotter and it gets drier. You tend to have a lot greater chance for a drought in July than in June, and that risk is just really important to avoid. But in small grains, we know in general they don't fill the seed well under hot nights, and our nights here can be overnight low of 80 or 85 once we get into July. And so just knowing what we know about cool season grass physiology, that's probably not a good match. So it's good to get them to mature faster. Ergot using Kansas varieties in Minnesota. I don't think it's any worse necessarily. I think ergot is also really environmentally driven. A whole lot as to how good pollination you get. If you get really good pollination, if you get bad pollination conditions, a lot of moist weather during that time period, you can get a lot of ergot no matter where you are. But it's heavily driven by environment. It's been difficult to breed for ergot resistance, even in rye, where they've spent tremendous amounts of resources on breeding for ergot resistance. It comes down a lot to just timing of when it flowers relative to what the weather's like, unfortunately.
Did Lee capture Catherine's question about earlier maturing varieties doing well in Minnesota?
Oh, I did not. I think it remains to be seen. As we saw, 801 did pretty darn well in Minnesota. So in that 1 year trial. So I don't think it's not necessarily bad. It's not necessarily better. We'll see.
Yeah, it makes sense. You're developing a lot of other positive traits that go along.
Right. Exactly. Yeah.
Hey, hello, everyone. The title of my presentation is Wheat and Seaweed Grass Hybridization and Ignore the Breeding Approach to Perennial Wheat. Since 2001, Lee and I have made many crosses between different wheat and wheatgrass species. So far, many advanced lines have been released. Among them, Durham wheat and Kansas hybrids have shown greater perenniality. Jake has tested some advanced line in Minnesota. A few of them lived for 3 years. Another type is hybrids is between bread wheat and seaweed grass. We test in our field for many years. They are among they had the highest grain yield, but the plants can only live for 1 year. Recently, one hybrid has done pretty well in Australia and Mexico. The plants lived for 2 or 3 years. Today, I will talk about this hybrid to show you a broader picture of perennial wheat breeding. The information I want to convey is that high yielding perennial wheat is possible. Weak perennial in one environment can be more perennial in another environment. Seaweed grass is originally from Western Europe. It has become an aggressive species in Australia and California along the coastlines. You can see this picture we grow in our field. The plants cannot survive the cold winter. The plants can only live for 1 year in Kansas. It's a tetraploid species with 28 chromosomes in the plants. It has J1 and J2 subgenomes. J1 is related to the E genome in Psilopyrum elongatum, and J2 is related to the EB genome in Psilopyrum basicum, and the V genome in intermediate wheatgrass. Professor Wang got a hybrid from me many years ago and did some investigations. The results showed that seaweed grass carry a lot of useful genes for the improvement of annual wheat. The plants are highly tolerant to waterlogging, magnesium toxicity, salinity, no nitrogen, and possible to heat. Also, the plants are resistant to wheat streak mosaic virus, Fusarium head blight, and the stem sow flies due to the solid stems. He and his team has created a draft genome assembly for seaweed grass and developed made addition lines for the transfer of useful gene into annual wheat. KSU is also doing genome assembly. The reference genome will be available sometime later this year. After the reference genome is available, they will develop a scheme sequencing analysis pipeline to assist the selection of seed wheatgrass hybrids. Here is the advanced line we released in 2016. Called B1107E. Durum wheat, the pedigree is that a durum wheat is crossed with seed wheatgrass first, then crossed with an unknown plant. We have tested this advanced line in our field for several years. It showed many positive traits. The plants ripen more naturally from top to bottom. Very vigorous regrowth. You can see in the middle picture. The maturity is very early. It only is about 10 days later than local annual wheat cultivars. In many experiments, this line has showed highest grain yield. The heads are easy to thresh. Also, the plants are resistant to most foliar diseases. This is one experiment. The group of lines is on the left. Are developed by us. Next is 4 lines were developed by Washington State University, former Soviet Union, Oklahoma State, and China. The last on the right is a local bred wheat cultivar. You can see this one, B1107, has the highest grain yield among all the perennial wheat lines. The grain yield was about 88%. Of local bred wheat cultivars. This experiment was conducted in the field with very low soil fertility. KSU has examined this line by means of scheme sequencing analysis and GISH. In the graph on the left, you can see all the wheat chromosomes have been mapped out. Then 2 wheatgrass chromosomes, S3 and S4, are mapped. This line has 56 chromosomes 56 chromosomes. So you can see on the right fish slide. You can see those pink color chromosomes. And the green color is chromosomes. Are from wheatgrass. Those pink chromosomes cannot be identified by a scheme sequencing analysis. This means these chromosomes are from wheatgrass, very different from those chromosomes in intermediate wheatgrass. We did this line is more like a soft wheat. The flour can make many products like bread wheat, muffin, cookie. The quality is just okay. Not as good as those hard winter wheat. We tested this line in our field for many years. The plants can only live for 1 year. But in Australia, there are plants in the field for 3 years. The first year grain yield was over 50% of local wheat cultivars. The second year grain yield was not made due to severe hail damage. The third year grain yield hasn't come out yet. At Summit, the plants are now in their second year. The first grain yield was about 60% of local wheat cultivars as shown in this figure. this blue bar is perennial wheat. One reason for lower grain yield is lower you see seed weight because this line is was planted very late in the half, just for you see and in the national you see conference. If this line is were planted at the normal time, I would expected you see bigger seeds and higher grain yield. You can see the first year you see plants that are heading. Then you see the second year you see plants. Most of the plants are still alive. Okay, so we have another line is derived from durum wheat and the seaweed grass hybridization. The plants showed very robust regrowth in greenhouse. As you can see in the bottom right, you see a picture, very vigorous regrowth. But in the field, the plants died quickly you see, in the heat. This line is very hard to thresh and tend to outcross you see, with neighboring you see, plants. So in summary, seaweed grass has many useful traits, which the high tolerance to waterlogging and salinity appear to be not possessed by intermediate wheatgrass. A weak perennial in one environment can be more perennial in another environment. B1107E has lived in Australia and Mexico for 2 or more years instead of only 1 year in Kansas. This suggests that environment makes a big difference for perennial wheat. More trials will be conducted along with the researchers in Australia and CIMMYT. Also, we will you see, do more work on this type of hybridization in the future. Okay. Thanks. That's you see what I have for today.
I might have missed this, Shuwen, but did you say if it's selfing or out pollinating?
Or the last you see advanced line tend to be cross pollinated, but the first line is I think is self pollinated, like just like you see annual wheat.
Thanks.
There was a question in the chat from Peter. So you talk about waterlogging, but how tolerant of drought is, I'm assuming it's a seaweed grass?
Oh, this one I don't know. I think it's not as drought tolerant as you say Kanza.
I just have a question about, so your comment about differing perenniality based on different environments. What do you think is your gut feel on what's causing that differences in perenniality? Does Kansas just have a really harsh winter that's causing issues, or what's your gut feel on what's going on?
Kansas has a harsh summer and a harsh winter, both. So the first line is spring has a spring growth habit. So it cannot you see survive the harsh winter in Kansas.
Makes me wonder. We have a small but growing winter durum breeding program, and we're still not to the point of having reliable winter survival in Minnesota, but our lines are way better than your average commercial spring durum. So that could be an interesting idea to cross some of our winter durums with the seaweed grass and see if we can bring in more winter hardiness that way.
Yeah, I agree. You see, those advanced line Jake tested. You see, crosses between the winter durum and the Kanza.
Okay. Well, if you ever want to access, further improved winter durums, don't hesitate to reach out. We'd be happy to share that for breeding purposes, I'm sure.
I think Jake has has obtained using some latest using line you see from see you see what you see as they look like you see in his test.
Winter terms for winter party, is that the idea?
One thought is, and I think you're alluding to this, but you know, the possibility of this type of perennial wheat being tolerant of less favorable conditions, I think is pretty exciting. We get a lot of questions or assumptions made about Kernza of, oh, you know, it's a hardy perennial, you can put it on some pretty poor ground, or you can use it in a buffer area next to a stream or a lake, and you know, such good nitrate uptake, isn't it going to be good for that use? And unfortunately, neither of those are really true. We don't find good performance in poor soils, and we know that it does not like to be in wetter environments. It sounds like this could thrive in those wetter environments and still function as a crop, which would be very valuable for our nitrate mitigation strategies. The other piece of it is the salinity tolerance, not too much of an issue in Minnesota, but you go to parts of North Dakota and there's large areas of land that are degraded and almost unfarmable at this point because of high salinity. I'm sure it's even worse in other parts of the West. So yeah, it seems like could be some really exciting applications out there in really niche areas, but in, you know, which usually being really niche is not necessarily a good thing, but in this case, they're niches that are hungry for a solution. So yeah, very, very interesting to see that. And so I guess my follow-up question is, and you mentioned collaborations with Jake, but I'm wondering if some of these particular seagrass hybrids, sea wheatgrass hybrids have come up here for testing in Minnesota.
I don't think this line you see will survive the cold winter but in Minnesota. But you see, I will set some seeds.
Doesn't hurt to try.
Yeah. Yeah. Also, I'm wondering if you see we can make some new crosses with some wheatgrass, sea wheatgrass plant from northern California. Then you see hybrids might be more cold tolerant.
And you know this better than me, maybe Jake does, but the new trials starting this year are going to include a lot more germplasm also, aren't they?
Yeah. Yeah. They just Jake's not here anymore.
No. I know, I'm like pointed in the direction where he was sitting.
Yeah, if the short answer is if there's enough seed of like to do a full like plot, we could try some. I'll try some.
Yeah.
I don't want to speak for Jake. Oh
yeah, I don't either. Yeah, and I don't want to. And I only know kind of tangentially also. Like this was part of a big conversation that happened. Yeah. You know, at the international meeting at Stoll, also that like lots of maybe you know more about this also. Like there's a bigger effort across many institutions to swap perennial wheat germplasm tested.
That's great.
So just full disclosure, I'm a rural sociologist, so I'm not a plant breeding out, but so I have a question when we're thinking about, I was sort of thinking of the earlier presentations about trying to figure out how to increase yield as like, you know, that's the goal that's being chased with the breeding programs. And then just wondering about with all the overproductions that's going on and all the other grains and commodities that are crashing the prices so that farmers can't make a living doing this, like do we need to be weaving in some of the other things too, which maybe we are, like I'm thinking of Kernza with the water quality benefits, like how do we track the ability for the different varieties of Kernza to play that function, and is that in like the traits that are being measured right now in the Kernza breeding programs?
Not directly. Oh, go ahead, Lee.
I was telling a grower just recently that I've got the solution for overproduction for you, but for some reason no one's interested. So, you know what often happens when we get overproduction is it becomes an arms race and everyone's just trying to produce more than the next guy because that's the only way to make it. There is openness often though at those times to try something new, because a lot of people say, well, if I can grow more wheat, but I just lose more money the more I grow. So maybe it's worth trying something new. So there is that opportunity. But we need to have a market, and that's really I think where Kearns struggles right now is having the market to be able to plant more of it. The yields are okay for going forward, I think, in general. But if we want to get more acres, it's going to have to be able to not have a huge premium price on it, and the way to get that price affordable for consumers is to get the yield up more than it is right now. So that it still is kind of one of the missing links, and it's not one you solve in a couple of years. It's a decades long solution, so we got to work on it today to get yields 10, 20 years from now, and we have to do the work today. So yield is the one that we gotta do the slog on, but the market is the thing that hopefully can be solved shorter and faster, and your expertise will hopefully help us there, because it's really the big one.
I think your question gets very much at the systemic issues that we all are operating under, I guess.
But we do have, I don't know if it's finally closed out or not, because it's been going for a while, but experiment that uses mini rhizotron tubes, so cameras that go in the soil to measure root characteristics, to make sure that we're not losing those root benefits as we breed for the agronomic and economic traits that are visible to the naked eye, right? Or easily measured. And so I don't think anyone has cracked the nut yet on paying farmers for ecosystem services in any kind of comprehensive way. So it's hard to say what the path is for that, but we do want to, our whole value proposition, what's different, some health value proposition too, right? But the big one is we're improving the environment in those ways. So we can't lose those outcomes, those benefits. And probably in addition to that mini rhizotron work, we ought to every 5 years or something, evaluate the latest varieties and say, okay, they're still here. We're still getting, nitrate reduction, just to, allay any potential concerns out there. Yeah, it's a good point that it's a whole system.
And if that market is being developed at the same time that the grain, like the use of the grain in the food or, something is, then it's two market enterprise functions that could be paying farmers or paying landowners.
Right. We kind of do it in a backdoor, right? Like a CSP payment is an ecosystem service payment.
Yeah.
The flip is similar. So Colin has said that the value of Kernza really ought to be seen as 1/3 grain, 1/3 forage, 1/3 ecosystem services that are going to come right now in the form of government payments, which don't last forever. And so it's not a forever solution, but for somebody to try it out, you can get a CSP contract for Kernza, you're not going to lose money. They might not get rich if you aren't able to sell it or whatever, but those are lucrative contracts. And if you can graze it, you're definitely going to be, or use the forage in some productive way, you'll be in good shape, and then the grain becomes just gravy on top. Now it's hard for farmers to line all those pieces up. It's what we've learned. Not a lot of people have livestock, are able to use it in that way. And then the NRCS programs, you have to apply like a year in advance, and it's like 2 years ahead of when you might want to sell the grain, just with this market changing as fast as it does. Yet, you're not trying to tear them. So it's hard to do, but I do think I agree with you that thinking about the multiple income streams is really important. I had one more question for Lee. First of all, Lee, you had the back-to-back slides of showing how good 1102 is, and then showing the maturity. And if you just lay those on top of each other, it's like the same pattern. It's amazingly consistent. So that's just kind of amazing. But my question is then, I think the next slide or the one after, you mentioned variety candidates, and it went into even numbers, 12, 14, 16, 18. So are you just in the interim ones as, advance the generation, and then only every other one you're actually looking at variety candidates at this point?
Yeah, that happened because as I've gone to the genomic selection 2 cycles a year, I basically only have a chance to create new varieties out of the even numbers for the most part, because the odd numbers, they're in the spring and there's no opportunity to really try those plants in the field. And so I've mostly not done it. I've generally taken plants from the greenhouse, put them in the field in the fall, trial them at least one winter as part of the developing the genomic selection model, and then use that data to pick out the parents of the variety candidate. That's how I get the even numbers. But it's a little difficult to resist sometimes the best plants, even if you've never evaluated them in the field. And people have grown 704. I think it's part of the Kernza cap, maybe. That particular selection was only made by genomic selection. They were the best plants from genomic predictions, no field trial of those plants at all. And they turned out to be, as good as the plants that were field trialed for the performance. So the genomic predictions are pretty darn amazing at times. And just one, year to year variation. If you evaluate one plant in the field for 1 year at one location and pick it as a parent because of that, well, there's so much genotype by environment interaction that one piece of information is not all that much more useful than having this genomic prediction model that's been developed over 25 different environments. So the predicted performance across lots of environments gives you probably more useful information about a plant's potential than its actual phenotype in one location. So
yeah,
maybe going forward, I'll start making ones in the spring too. It's just that it's hard to get them intermated in the summer. So by that time in the fall, and I'm to the next generation. So I'm on the go. So yeah, my goal is to make at least one new candidate every year out of every other generation, maybe more, but I'm running out of isolations, right? Every time you make a new cross, you got to grow seed in isolations, and I've got little isolations all over our property now. So I know Praveen packed them a little closer and planted corn on them. You can do that too, but I've tried to kind of keep them separate.
Yeah, really interesting. Thank you.
Sorry, follow-up question from Walid about Mitch's comment about rhizotrons. Are there any intermediate wheatgrass vintage trial studies that examine whether breeding for higher yield is not taking place at the expense of environmentally friendly traits, for example, water removal?
No.
There
aren't any studies that are answering that exact question.
Do we have in the mini rhizotron study, do we have like rush or C4 material, any of the older material? It was just a breeding.
Yeah, so the rhizotron study is embedded within cycle 6 of the breeding population. So it's not sampling over time, but that's a good point of it'd be interesting this is something we talked about. It'd be interesting to see, the difference in root architectures.
How it's changed.
I can tell you anecdotally that for many years, from the beginning, we always throw the plants out of the greenhouse, right, when we're done with them. So you rip them out of the pot and you see what the roots look like. And definitely the current generation, 18 cycles in, there's not as much root boundness in those plants as there used to be. So I think they're becoming less root producing. And the question is, for a service like nitrate uptake, you don't actually need near as many roots as most plants have, because water moves to the soil, it'll move to the root, it doesn't really matter. And for phosphorus uptake, we have some evidence that we're making the plants more responsive to mycorrhizal introduction, which might be through having smaller roots and making up for it with more mycorrhizae. The mycorrhizae might actually be better at creating soil carbon than root degradation is. So maybe smaller roots, more mycorrhizae is actually going to give us more soil carbon and just as good as soil nitrogen uptake. It's hard to make the extrapolation from root mass, but the differences between a Kernza plant with 10% less roots versus a wheat plant with very little roots there for only a few months of the year, those are qualitative differences. So the tiny changes we're making in root biomass relative to the change of a plant that's there all the time and rooted several meters down, it's still clearly in a different category. But it's a fair question that things are going to change, and they might change for the better or change for the worse. With the whole mycorrhizal thing, generally, when people say you breed for yield, you reduce mycorrhizal responsiveness, you make them just dependent on fertilizers and whatever. in our case, they're becoming more mycorrhizal responsive as we breed for yield. So, yeah, you can't make assumptions about what's going to happen, but we know change will happen. It's a matter of what scale it is, what the magnitude is.
It's interesting that you anecdotally see that reduction in root mass. My simplistic understanding of some of the results from the mini rhizotron study is that we at least have not seen a hard trade-off between above ground productivity. Yeah. So from that perspective,
right,
it's unlikely we're selecting in a really negative way against root biomass, but we're certainly not selecting for it at this point. Yeah. So it could easily just drift away, if nothing else.
I know Praveen was also excited. This is a different thing, but he was also excited about maybe doing EMS on intermediate wheatgrass. Did he ever end up doing that?
Yeah.
Okay. and what's kind of the status of that?
So I've done EMS as well. I had a horrible time finding anything out of it. So it's really hard to differentiate new variation that you just made versus all the variation that's already there, unless you use a molecular approach. And I've been a partner of an experiment in Denmark where they actually do sequencing to identify the new mutations that are causative in the genes of interest. And they found direct causative mutations through EMS in, for instance, the Q gene, the major domestication of wheat gene, and others. So there is potential to do it, but doing it as I did it and just throwing some EMS on and looking for new variation, probably not going to happen very well, because especially those really major genes have to be they're recessive often, and so you have to get them into a homozygous state, which wheatgrass doesn't like to be homozygous, so you have to force that. And it's often a hexaploid, and so there's 3 genomes in there, and if it has to be recessive, you might have to mutate 6 copies and then make crosses to paste it all together into something that gives you the phenotype you're looking for. So I think directed mutation it has great potential, but it has to be done with a big investment genomically to actually understand what you're doing and then assemble the target genotype and hopefully phenotype. That program has ended, and they're going to be sending, if we can get the details worked out with the inspections, they're going to send me the seeds that were developed through that directed mutagenesis project, and I'm going to do field evaluations of those. They've only had them in the greenhouse, so they don't actually know what the difference is in the field, but they definitely have seen phenotype changes in the plants that are indicative of the kind of thing they would be looking for with a change in the target genes.
Did they do transcriptomics on it then to get the genotype data?
No, it's they have basically a patented procedure. It's called a find it thing. It's based on DNA sequencing only, and they pool the DNA in various different ways so that you essentially make a library of 1 million different plants that you can then interrogate and find the target mutation at low cost. There's a nice paper published called "Find It," a trademark, and describes their methods very, very clearly.
So the EMS population here at the U, the goal of that was to find a self-pollinating. So on the EMS population, we put out a bunch of just pollen isolation bags to see if we get any self-seed.
Did you find anything? Did you find anything?
Which, hearing what Lee just said, it's that makes sense why we probably did.
The whole selfing thing, it's like there's a lot of other reasons why an outcrossing plant doesn't self besides self-incompatibility. Basically, lethal genes. And I did a lot of work, 5 years of pretty intensive work on self-pollinations to try to make hybrids, inbreeding repeated generations, and basically concluded that by the 3rd generation you've accumulated so many lethal recessives that doesn't matter about the self-incompatibility mechanism. None of the progeny make it anyway. So and that's largely what's happening. Even if you can overcome the self-incompatibility by genetically, they're going to be non-viable because of inbreeding depression.
And then Wally just responded to your comment, Lee's root mass can decrease, but maybe root and canopy conductances are moving in the favorable direction, though probably something worth checking out.